Welcome to this presentation on vaccine pharmacovigilance signal detection. In this session, we will explore the critical frameworks and statistical methods used to identify rare adverse events following immunization, especially within the pediatric surveillance systems. Vaccines are among the most effective public health interventions in history, but continuous safety monitoring remains essential to maintain the public trust and ensure early detection of potential risk. We will discuss signal detection metrics, pharmacovigilance databases, disproportionality analysis, casualty assessment, and real world surveillance strategies used globally. By the end of this presentation, you will understand how modern vaccine safety systems detect, evaluate, and respond to the rare adverse events in large populations. So let's begin. Detecting vaccine safety signals is one of the greatest challenges in modern pharmacovigilance. Some rare adverse events are extraordinarily rare, occurring as infrequently as one case per billion vaccine doses administered. This creates a major surveillance challenge. How do we identify a true safety signal hidden within millions of normal outcomes? Passive surveillance systems such as VAERS and VigiBase rely on large scale reporting patterns to detect the unusual clustering of events. In many cases, more than 200 independent reports may be required before statistical algorithms recognize a potential signal worthy of the investigation. This means that the rarest adverse events demand the most sophisticated epidemiological tools, massive datasets, and continuous global monitoring. In vaccine pharmacovigilance, finding a single meaningful signal often requires analyzing millions of data points. In pediatric vaccine pharmacovigilance, complexity increases dramatically because children are not simply small adults. The first challenge is developmental variability. Background medical event rates change rapidly during the first 2 years of life, making it difficult to distinguish coincidence from causality. Second, co administration complicates attribution. Infants often receive multiple vaccines during a single visit, making it challenging to determine which product may be associated with a reported adverse event. Third is reporting bias. Highly informed or anxious parents may overreport mild symptoms, while passive surveillance systems still capture only a small fraction of total adverse events, sometimes as low as 1 to 10%. Finally, there is ethical pressure. False positive safety signals can rapidly undermine public confidence in vaccination programs worldwide, even before causality is scientifically established. For because of these factors, pediatric pharmacovigilance requires exceptionally careful statistical interpretation, clinical judgment, and transparent communication. In this scenario, a national surveillance database is evaluating the safety profile of a newly introduced pentavalent vaccine after post license rollout. The database currently contains 10,000 adverse events following immunization reports. Among these reports, 87 cases of febrile seizures were identified in children younger than 2 years of age. The most important question is whether these observed number exceeds the expected national baseline incidence, which is approximately 0.6 cases per 1,000 doses administered. At this stage, the signal is not automatically considered casual. Instead, it becomes a statistical alert requiring further pharmacovigilance analysis. The primary objective is to determine whether the febrile seizures are occurring at a genuinely higher than expected frequency or whether the observed cluster can still be explained by the background coincidence, reporting patterns, or random statistical variation. This is where advanced signal detection metrics and epidemiological frameworks become essential in the modern vaccine safety surveillance. Now comes the critical decision for the pharmacovigilance analyst. Which statistical method should be used to investigate the signal? With over 10,000 surveillance reports available, there are multiple analytical pathways. The first method is proportional reporting ratio. PRR. This approach compares how frequently a specific adverse event is reported for one vaccine versus all the other vaccines within the same database. It's fast, practical, and widely used for signal detection. The second method is observed versus expected analysis. Here, analysts compare the number of reported events with the number statistically expected in the general population using epidemiological background rates. This method is especially valuable for identifying whether the event truly exceeds normal coincidence. The third approach is sequential probability ratio test or SPRT. Unlike static analysis, SPRT works in real time as new reports continue to arrive, making it highly effective for continuous national vaccine monitoring. Each method answers a slightly different question, and choosing the correct one can determine whether a potential safety signal is confirmed, dismissed, or escalated for urgent investigation. The danger of the incomplete approach. Now, let's talk about one of the biggest problems in the vaccine safety signal analysis relying only on the PRR without understanding the bigger picture. At first glance, PRR or proportional reporting ratio may seem powerful. A high PRR value can look alarming and may appear to suggest a strong association between a vaccine and an adverse event. But this is exactly where the misinterpretation begins. The first major issue is the absence of the background rates. Just because an event is reported more frequently after the vaccination does not automatically mean the vaccine caused it. Many medical conditions naturally occur in the population every day. For example, heart attacks, strokes, seizures, or autoimmune disorders already happen at baseline levels. So, even if the PRR is greater than 2, the observed event rate may still actually be lower than what is normally expected in the general population. Without comparing against epidemiological background data, conclusions can become misleading. The second problem involves the database composition. Imagine a situation where multiple vaccines in the reporting database are associated with the same adverse events. In that case, the denominator of the PRR equation becomes inflated. This dilution effect can hide genuine safety signals or distort the apparent strength of the associations. So, PRR heavily depends on the structure and composition of the database being analyzed. The third issue is multiple testing inflation. Modern pharmacovigilance systems analyze thousands of vaccine event combinations simultaneously. Statistically, when you test huge numbers of associations, some signals will appear significant purely by the chance. This is known as alpha inflation or false positive signal generation. In other words, some detected safety signals may not represent real biological risk at all. They may simply be statistical noise. So, the key takeaway from this slide is that PRR should never be interpreted in isolation. It is only screening tool, not definitive proof of causality. Proper vaccine safety assessment always requires clinical evaluation, epidemiological studies, biological plausibility, and contextual interpretation. A signal alone is not evidence, it is only the beginning of the investigation. The gold standard signal triangulation. After understanding the limitations of relying on single metric like PRR, we now move forward what is considered the gold standard in the pharmacovigilance and vaccine safety science. Signal triangulation. Signal triangulation means combining multiple analytical approaches instead of depending on just one method. Because vaccine safety is complex, no single statistical tool can provide complete certainty. Each method answers a different scientific question, and together they create a much stronger and reliable assessment. The first component is the PRR or proportional reporting ratio. This acts as initial screening tool. It helps identify whether a particular adverse event is reported disproportionately within the database. In simple terms, PRR tells us is this event appearing more frequently than expected compared to the other events in same reporting system. But remember, PRR alone only detects a potential signal, it does not confirm the causality. The second component is O over E analysis, meaning observed over expected epidemiology. This method compares the number of observed adverse events after vaccination with the number that would normally occur in the real world population background. This step is extremely important because it introduces real epidemiological context. It helps answer the question, are we truly seeing excess cases beyond normal expectations? This serves as a validation layer for the signal. The third approach is SPRT, sequential probability ratio testing. Unlike static analysis, SPRT is dynamic and time based. It continuously monitors incoming data in real time and evaluates whether a safety threshold can be crossed. This is especially useful during mass vaccination campaigns where rapid detection of emerging safety issues is critical. In other words, SPRT functions as an active surveillance and monitoring tool. The fourth component is EBGM or empirical Bayesian geometric mean analysis. This method adjusts for statistical noise and instability, especially when dealing with the rare events or small sample sizes. Bayesian adjustment reduces the likelihood of false positive signals caused by random fluctuations. EBGM essentially asks, after correcting for noise and uncertainty, how strong is the signal really? This acts as a confirmation layer. When all these methods point in the same direction, confidence in the signal becomes much stronger. But if only one method shows concern while others do not, investigators must interpret the findings cautiously. So, the major lesson from this slide is simple but powerful. No signal metric carries the full weight of the vaccine safety assessment. True pharmacovigilance depends on triangulation evidence from multiple complementary methods before drawing conclusions. In vaccine safety science, confirmation comes from convergence, not from the one isolated number. Internal mining, disproportionality, and Bayesian metrics. Now let's move deeper into the actual statistical methods used in the pharmacovigilance databases for internal signal mining. In this slide, we compare two of the most important analytical approaches used in the modern safety surveillance system, PRR, which is a disproportionality metric, and EBGM, which is a Bayesian metric. Both aim to detect potential vaccine safety signals, but they work in very different ways. Let's begin with the PRR or the proportional reporting ratio. PRR is built using a 2 by 2 contingency table. Here, the variable A represents report specific adverse event associated with the target vaccine. B represents all other adverse events reported with that same vaccine. C represents the same adverse event reported with other vaccines. And D includes the adverse events from all other vaccines. Using these values, PRR compares the proportion of a specific adverse event in the target vaccine against the proportion seen in all other vaccines within the database. In simple terms, PRR asks the question, is this adverse event being reported disproportionately or more often with this vaccine compared to others? To determine whether the signal is meaningful, researchers commonly use the Evans criteria. According to this threshold, a primarily signal is considered significant when the PRR is greater than or equal to 2. The chi-square value is greater than or equal to 4 and at least 3 cases are reported. However, PRR has an important limitation. It can become unstable when dealing with small numbers or rare events. A few reports may artificially inflate the ratio and create false positive signals. This is where EBGM becomes extremely valuable. EBGM stands for Empirical Bayesian Geometric Mean. Unlike PRR, EBGM uses Bayesian statistical modeling to adjust for random noise and variability, especially in sparse datasets. Instead of relying solely on the raw observed counts, it estimates a posterior distribution of the signal strength. This graph shows here represents that probability distribution. The key parameter in EB05, which is lower fifth percentile confidence bound to the estimated signal. Why is it important? Because EB05 provides a conservative estimate of the signal strength. Even after counting for uncertainty and statistical fluctuation, if the EB 0.05 value remains greater than or equal to 2, the signal is considered robust and potentially meaningful. In fact, this threshold is widely used in FAERS pharmacovigilance system. So while PRR acts as a rapid screening tool, EBGM provides a more stable and reliable confirmation method, especially for rare adverse events and small diseases. The major takeaway from this slide is that disproportionality methods detect possible signals quickly, while Bayesian methods help to refine those signals by reducing the statistical noise and improving reliability. Together, they form a crucial foundation for modern vaccine safety signal detection. Now that we have explored internal database mining methods like PRR and EBGM, the next step is to place safety signal into real-world epidemiological context. This slide introduces two extremely important approaches used in the advanced vaccine safety surveillance. O over E analysis and sequential probability ratio testing or SPRT. Let's begin with the O over E analysis, which stands for observed versus expected analysis. The central idea behind this method is very straightforward. We compare the number of adverse events actually observed after vaccination with the number of events that would normally be expected to occur in the population by chance alone. The formula shown here calculates this relationship by dividing the observed number of cases by expected number of cases, where the expected count is estimated using percent time exposure multiplied by the known background incidence rate from the general population. This is one of the most important epidemiological validation methods because it helps answer a critical question. Are we truly seeing more cases than would normally happen naturally? For example, conditions like myocarditis, seizures, strokes, or autoimmune disorders already occur at baseline frequencies in the society. During mass vaccination campaigns, some of these events will inevitably occur shortly after the vaccination simply due to coincidence. O over E analysis helps separate the coincidence from a potentially meaningful excess signal. The histogram shown on the left represents the expected distribution of the events, while the red bar represents the actual observed number of cases. If the observed value lies significantly above the expected range, investigators may suspect a potentially safety signal requiring further investigation. Importantly, this method often uses exact Poisson statistical testing and confidence intervals to determine excess is statistically significant. Now let's move to SPRT. Unlike traditional analysis that evaluate data only after collection is complete, SPRT works continuously in real time. As new adverse events report accumulate, the system repeatedly updates like likelihood ratio and checks whether a predefined signal threshold has been crossed. The graph on the right demonstrates this concept. The line fluctuates over time as cumulative reports increase. Once the statistical threshold has crossed, a potential safety alert is triggered. This approach is especially valuable during large scale vaccination programs because this allows early detection of emerging risk while still controlling false positive errors. One major advantage of SPRT is speed. Instead of waiting months or years for final analysis, health authorities can identify unusual patterns rapidly and intervene sooner if necessary. In fact, systems such as US Vaccine Safety Data Link use SPRT based continuous monitoring to track the vaccine safety in near real time. So, the key message is that internal statistical signals alone are not enough. Vaccine safety assessment must also include external epidemiological validation and dynamic real time monitoring. O over E analysis tells us whether events exceed real world expectations, while SPRT tells us whether the signals are emerging continuously over time. Together, they provide a much stronger and more clinically meaningful framework for vaccine pharmacovigilance. Step by step, the pentavalent case study. Now, let's bring everything together using a practical example. A hypothetical pentavalent vaccine safety case study. This slide demonstrates how multiple pharmacovigilance methods work sequentially to evaluate whether a detected signal may represent a true safety concern. We begin with the initial input data. In this scenario, there were 87 reported seizure cases following administration of approximately 4.2 million vaccine doses. The known background incidence rate for seizures in the pediatric population is 3.2 cases per 1,000 child. The first step is calculating the expected number of cases. Using the background incidence rate, the amount of person time exposure, the model estimates that only about 1.55 seizure cases would normally be expected to occur naturally during the observational period. This brings us to note 2. The observed versus expected analysis or O over E ratio. Here we divide the observed number of cases 87 by the expected number 1.55. The resulting O over E ratio is 56.1 with a 95% confidence interval ranging from 44.9 to 69.3. This means that the observed seizure count is dramatically higher than it would normally be anticipated based on the background epidemiology. At this stage, a strong epidemiological signal is detected. Next comes the note 3 PRR analysis. Using disproportionality analysis, the PRR value is calculated 5.17 with a chi-square static of 112 and a sample size of 87 reports. This clearly exceeds the standard events criteria thresholds using in the pharmacovigilance, where PRR greater than or equal to 2, chi-square greater than or equal to 4, and at least 3 cases indicate a preliminary safety signal. So now both epidemiological validation and the disproportionality analysis are pointing towards the same conclusion. But the analysis does not stop there. Note 4 introduces Bayesian confirmation using the EB05 analysis. After adjusting for statistical uncertainty and random noise, the EB05 value remains 3.9, well above the FDA threshold of 2.0 for a robust Bayesian confirmed signal. This is important because Bayesian adjustment helps ensure that the signal is not simply the result of rare event instability or small number fluctuations. At this point, all major analytical layers are aligned. Observed versus expected analysis shows incidence, PRR shows disproportionality, and Bayesian adjustment confirms the statistical robustness. This is exactly what high quality signal triangulation looks like in the modern vaccine pharmacovigilance. The key lesson is that no signal or single calculation determines the vaccine safety conclusions. Instead, investigators move step by step through multiple analytical checkpoints to determine whether a signal is biologically and statistically credible. In real world pharmacovigilance, strong evidence emerges when multiple independent methods converge on the same finding. The method comparison matrix. After exploring each pharmacovigilance method individually, this slide brings them together into a direct comparison matrix. The purpose of this comparison is to understand that every vaccine safety detection method has unique strengths, limitations, and ideal use cases. No single approach is universally superior, each contributes a different layer of evidence. Let's start with the first row data requirements. O over E or standardized incidence ratio analysis requires external epidemiological baseline data. This means it depends on real world background incidence rates from the general population. In contrast, PRR and EBGM are internal database mining methods. They work entirely within the pharmacovigilance database itself and do not require external baseline incidence information. SPRT, however, depends on the population exposure data because it continuously monitors the accumulating reports over time in relation to the vaccinated populations. Now, let's examine the ability to handle rare adverse events. Rare event analysis is one of the biggest challenges in the vaccine safety surveillance. PRR often struggles with the rare events because small numbers can create unstable or inflated ratios. On the other hand, O over E analysis, EBGM or SPRT perform much better in rare event settings. EBGM is particularly strong because Bayesian adjustment reduces random statistical noise associated with the sparse datasets. Next is real-time use capability. Among all the methods, SPRT stands out as primary real-time monitoring tool. It continuously evaluates the incoming data and rapidly identifies the emerging safety signals. The other methods O over E, PRR, and EBGM are generally more static analysis performed after data accumulation rather than continuous live monitoring. Now let's discuss bias vulnerability. every statistical method has potential weaknesses. O over E analysis can be affected by underreporting because passive surveillance system may not capture all adverse events. PRR is sensitive to denominator distortion, meaning the composition of the database itself can influence the signal strength. EBGM can also be affected by database composition, although Bayesian adjustment helps to reduce the instability. SPRT depends heavily on the assumptions built into hypothesis model and threshold specification. Incorrect assumptions may influence signal detection performance. Finally, the last row highlights the real world regulatory usage. O over E approaches are commonly used by the organizations such as WHO and CDSCO. PRR is widely used by agencies like European Medicines Agency and WHO for disproportionality signal screening. EBGM forms a major component of FDA FAERS pharmacovigilance system because of its Bayesian robustness, and SPRT is heavily used within the CDC vaccine safety data link for near real time surveillance monitoring. The central takeaway from this slide is that modern vaccine safety assessment is multidimensional. Different methods answer different scientific questions. PRR identifies the disproportionality, EBGM stabilizes the noisy signals, O over E validates the real world excess risk, and SPRT enables continuous real time surveillance. When these approaches are combined strategically, they create a far more reliable and scientifically rigorous pharmacovigilance framework than any signal or single metric alone. The architecture of surveillance India AEFI system. After understanding the statistical methods used in the vaccine safety detection, it is equally important to understand how adverse event surveillance is actually organized in real world healthcare systems. This slide illustrates the architecture of India AEFI surveillance systems. AEFI meaning the adverse events following immunization. India follows a structured multi-level reporting and review framework designed to detect, investigate, and evaluate the vaccine safety signals from grassroots level all the way to national regulatory authorities. Let's begin with the level 1, the peripheral health worker. This is the frontline level of the vaccine safety surveillance. Healthcare workers, nurses, vaccinators, and peripheral staff are often the first individuals to identify an adverse event following immunization. At this stage, the health worker documents the event using the official Form 1 AEFI reporting format and forwards this to the report to block or district level for further review. This level is extremely important because the quality of the entire surveillance system depends heavily on the accurate early reporting. If the adverse events are not recognized or documented properly at the grassroots levels, signals may never enter the surveillance pipeline. Next comes the level 2, the district immunization officer. Here, reports are reviewed, classified, and assessed for completeness. The district level authorities determine whether the event meets criteria for the serious AEFI investigation and then forward relevant cases to the state committee. This stage acts as the first formal filtering and coordination layer in the surveillance process. Level 3 is the state AEFI committee. At this level, detailed medical review and casualty assessment are performed. The committee evaluates clinical records, timelines, laboratory findings, vaccine storage conditions, patient history, and alternative explanations. Importantly, commonly uses the WHO casualty assessment algorithm during the stage to classify whether the event is consistent or causal association, indeterminate, coincidental, or unrelated to vaccination. This is one of the most scientifically critical stages because it shifts the process from simple reporting to evidence based causality evaluation. Finally, we reach the level 4, the National AEFI Committee. This is the highest surveillance and regulatory review body. At the national level, experts conduct signal review, trend analysis, policy recommendations, and coordination with regulatory authorities such as CDSCO, the Central Drugs Standard Control Organization. At this stage, broader national safety patterns are evaluated. If necessary, recommendations may include enhanced surveillance, label modifications, risk communication, or additional investigations. The pyramid structure shown in this slide demonstrates how local reports progressively move upward through increasingly specialized levels of analysis and oversight. The key takeaway is vaccine safety surveillance, which is not just about the statistics or algorithms. It is also about the organized public health infrastructure, coordinated reporting systems, expert medical review, and regulatory decision making. A strong AEFI system ensures that potential vaccine safety concerns are detected early, investigated scientifically, and addressed systematically to the CDSCO to maintain the public health and immunization safety. Standard signal detection workflow. Now that we understand both the statistical methods and the surveillance architecture, this slide brings everything together into a complete signal detection workflow used by the organizations such as WHO and CDSCO. This workflow represents the standard pathway through which a potential vaccine safety signal moves from initial reporting all the way to the regulatory decision making. Let's go step by step through the process. Step 1 begins with the data collection. This stage includes both passive surveillance and active surveillance systems. Passive surveillance involves spontaneous reporting of the adverse events by the healthcare professionals, hospitals, vaccination centers, or patients themselves. At the same time, active surveillance systems such as sentinel monitoring sites proactively search for the adverse events rather than waiting for reports to arrive voluntarily. The quality of signal detection depends heavily on the completeness and accuracy of this data collected. Step 2 is the case definition standardization. Here, standardized clinical definitions developed by the Brinton collaboration are applied to ensure consistency in identifying the adverse events. This is extremely important because the same clinical condition may otherwise be reported differently across the institutions or countries. Standardized definitions improve the reliability, reproducibility, and comparability of the vaccine data safety. Step 3 involves the disproportionality analysis. At this stage, statistical data mining methods such as PRR and information component are calculated using large pharmacovigilance databases like VigiLyze. The purpose here is to identify unusual reporting patterns that may represent the potential safety signals. However, these signals are still preliminary and require additional validation before conclusions can be made. That leads us to step 4 observed versus expected validation. Here, national epidemiological incidence data, often obtained from systems such as HMIS, are used to determine whether the observed adverse event frequency truly exceeds the expected background rate in the population. This step adds real world epidemiological context and helps to distinguish the genuine excess risk from the coincidental occurrence. Step 5 is the casualty assessment. At this stage, the committees apply the WHO casualty assessment algorithm to classify the event into categories such as consistent causal association, inconsistent association, indeterminate, or unclassifiable. This process considers factors like timing, biological plausibility, clinical evidence, alternative explanations, and existing scientific literature. This is one of the most critical stages because statistical association alone does not prove causation. Finally, step 6 is the signal decision making. After national level expert review, regulatory authorities determine whether the action is required. Possible outcomes may include enhanced surveillance, label changes, risk communication, restriction of vaccine batches, additional studies, or continued monitoring. This final stage transforms raw surveillance data into the evidence based public health action. The most important lesson from this slide is that the vaccine safety signal detection is not only a single statistical calculation, it is a structured scientific workflow involving the surveillance, standardization, statistical analysis, epidemiology, casualty assessment, and regulatory oversight. Every stage acts as a filter to ensure that the decisions are based on rigorous evidence rather than isolated reports or incomplete interpretation. This systemic approach is what allows the global vaccine programs to maintain both scientific integrity and the public trust. Now let's apply everything we have learned using a high yield exam style scenario commonly encountered in pharmacovigilance and vaccine safety assessments. In this example, a meningococcal conjugate vaccine was monitored over a 6-month period. Analysts identified a PR value of 3.2 for anaphylaxis with 9 reported cases and chi-square value of 6.8. At first glance, this appears significant because the events criteria of PRR are technically met. Remember, events criteria require a PRR greater than 2, chi-square value greater than 4 or equal to 4 and at least 3 reported cases. So basically purely on PRR one might initially suspect a potential safety signal. However, the Bayesian analysis tells a different story. The EBGM value is only 1.4 and most importantly the EB05 value is 0.9. Now, this is the critical detail. EB05 represents the lower fifth percentile confidence bound used in the Bayesian signal confirmation. In systems such as FDA, FAERS, a confirmed robust signal generally requires EB05 value greater than or equal to 2.0. Since the EB05 here is below 2.0, the Bayesian model suggests that the apparent PRR interval and the elevation may simply be due to the random statistical fluctuation or small number noise that rather can be a true safety signal. Therefore, the correct answer is option B. The signal is not confirmed because EB05 is less than 2.0, overrides the preliminary PRR signal. This question highlights an extremely important principle in the pharmacovigilance. PRR alone is not only sufficient to confirm a vaccine safety signal. Bayesian confirmation methods are used to determine whether the signal remains stable after adjusting for statistical uncertainty. Let's briefly review why the other options are incorrect. Option A is incorrect because meeting events criteria only indicates a preliminary disproportionality signal, not definitive confirmation. Option C is incorrect because SPRT is actually a major real time surveillance tool used in the vaccine safety monitoring systems like the CDC Vaccine Safety Data Link. Option D is incorrect because O over E validation remains essential even when a PRR signal is met or present. Real world epidemiological context is always required before drawing the conclusions. The major takeaway from this slide is that the robust pharmacovigilance depends on the signal triangulation and conservative confirmation methods. A statistical spike does not equally found the causality. Strong vaccine safety conclusions requires consistency across multiple analytical approaches. The golden rule of pharmacovigilance. As we conclude this discussion on the vaccine pharmacovigilance and signal detection, this slide summarizes the single most important principle in the modern vaccine safety science. The golden rule of pharmacovigilance is simple: never rely on one metric system alone. Vaccine safety assessment requires triangulation of the evidence using the multiple complementary methods. The first principle is to triangulate the signals. Whenever a potential adverse event signal appears, investigators should compare findings across multiple analytical approaches rather than depending on one isolated statistical result. A PRR signal is alone may represent a true association, but it may also reflect reporting bias, database distortion, random fluctuation, or statistical noise. That is why triangulation is essential. Consistency across independent methods increases the confidence in the signal's reliability. The second principle is validation using the O over E analysis. Even if a signal appears statistically elevated inside a database, we must always ask, does the observed event frequency truly exceed the normal background incidence in the real world population? O over E validation provides epidemiological context and helps distinguish the meaningful excess risk from the coincidental occurrence. The third principle is the Bayesian confirmation. Bayesian confirmation methods include the EBGM and EB05, which help to stabilize the signals by adjusting for uncertainty, sparse data, and the small number noise. This is especially critical in vaccine surveillance because rare adverse events can easily generate misleading disproportionality signals if Bayesian correction is not applied. That is why this slide emphasizes this core message. No single metric can carry the full weight of vaccine safety assessment. Never rely on PRR alone. Always verify signals using EB05 and observed versus expected validation before regulatory action is considered. This philosophy is what separates modern evidence based pharmacovigilance from simplistic statistical interpretation. Ultimately, the vaccine safety surveillance is not about reaching to isolated numbers, it is about integrating the epidemiological statistics, Bayesian modeling, clinical review, and casualty assessment into one coherent scientific framework. That integrated approach is what protects both public health and public trust. Thank you everyone for watching this session on vaccine pharmacovigilance and signal detection. I hope this lecture helped simplify these advanced concepts and made signal vaccine detection easier to understand for your exams, research, and clinical learning. If you found this video helpful, don't forget to like, share, and subscribe to support more detailed medical and pharmacological content. Thank you once again for watching, and I'll see you in the next lecture.